Hello all, myself Srinidhi Kulkarni, the author of the paper Enhanced Molecular Similarity Protein-Ligand Aligner for Accurate Feature Molecular Extraction from Protein-Ligand Interactions. So this is the table of these are the table of contents: abstract, introduction, problem statement, literature survey, methodology, results and discussion, conclusion, and references. So if I want to start with the abstract, protein-ligand interactions are fundamental in various biological and medical fields, influencing drug discovery and therapeutic development. In recent years, deep learning has revolutionized the study of these interactions, but significant challenges remain in accurately representing molecular structures for DL modules. Traditional featurization techniques often depend on handcrafted features, requiring expert knowledge and potentially missing crucial molecular aspects. The progress, the work addresses these challenges by developing and evaluating a novel protein ligand feature extraction system using an enhanced molecular similarity protein ligand aligner. So in short, we call it as EMSPLA. The primary objective was to leverage EMSPLA for similarity matching in protein ligand interactions, improving the predictive model accuracy. The methodology combined convolutional neural networks for local feature extraction with an attention module to capture long distance dependencies, enhancing binding site predictions. Using the PDBbind 2020 dataset, the EMSPLA model demonstrated superior performance with a root mean square error of 0.67, surpassing current state-of-the-art models. So we could see that in the conclusion part and results and discussion chapter. So these findings highlight the system's potential for efficient deployment and scalability. Positioning it is a powerful tool in computational biology and drug discovery, ultimately advancing our understanding of protein-ligand interactions. So if I want to discuss the introduction part, so a protein-ligand interaction involves a binding of a ligand, which is typically a small molecule to a specific site on a protein. Proteins are large, complex molecules that play many critical roles in the body, including acting as enzymes, signaling molecules, and structural components. Ligands can be various substances, including drugs, hormones, and or neurotransmitters. The interaction between a protein and its ligand is fundamental to many biological processes, as it can alter the protein's structure and the function, often triggering a biological response. Protein ligand interactions are crucial in various biological and medical fields. They can regulate physiological processes by activating or inhibiting the protein's function. For instance, in enzymatic reactions, a substrate binds to an enzyme to catalyze a biochemical reaction. In the pharmaceutical industry, understanding these interactions is vital for drug discovery and development. Drugs are designed to target specific proteins, modulating their activity to treat diseases. By binding to their target proteins, these drugs can inhibit or stimulate the protein's function, thereby altering the disease process. The specificity and affinity of the ligand for its protein target are key factors in determining the efficacy and safety of the drug. So this is about the introduction part. So in the recent year, deep learning has revolutionized the study of protein-ligand interactions, a subset of machine learning. The deep learning is a subset of machine learning, which uses the neural networks with many layers. So as it uses the many layers, it is called as deep to model the complex patterns of data. In protein-ligand interactions, deep learning models can predict binding affinities, identify potential binding sites, and even design novel ligands. So these models are trained on large data sets of known protein ligand complexes, learning to recognize subtle patterns that guard binding interactions. For example, CNN, convolutional neural networks, have been used to analyze 3D structures of protein ligand complexes capturing spatial features that are critical for binding, recurrent neural networks, RNNs, another type of DL architecture. So it have been applied to sequential data such as protein sequences to predict their interactions with ligands. Our significant challenge in leveraging deep learning for protein-ligand interactions is the need to accurately represent molecular structures in a form that the models can process. This is where the featurization comes in. A featurizer converts the complex multidimensional information of molecular structures into feature vectors, which are numerical representations that capture the essential characteristics of the molecules. So this is about the, still if you want to understand the introduction part, then if I want to tell regarding the introduction part, designing an effective featurizer is critical because the quality of the feature vectors directly impacts the performance of the real models. These features might include information about the molecular geometry, electronic properties, and the physiochemical characteristics of the ligand and protein. Traditional featurization techniques often rely on handcrafted features, which require expert knowledge and may not capture all relevant aspects of the molecules. However, DL models can be used to learn features directly from the raw data. This process is called as deep representation learning. This approach can uncover more intricate and informative representations that might be missed by manual featurization. As a result, designing a robust featurizer or leveraging deep learning to automatically extract features is essential for understanding of protein-ligand interactions and improving the accuracy of the predictive models in the drug discovery and other applications. So this is about the literature survey. 2020, a multi-channel substructure graph-gated recurrent unit, which consisted of various neural network having different attention modules, which were applied on these molecules, substructure for the predicting and learning different properties of the molecules. So what are the findings of this particular paper? So let us understand the findings. ESOL, so this particular paper, used the dataset of ESOL, PDBbind, and Protein-Ligand and free solvation and lipophilicity datasets. These were used, and correspondingly, the RMSE root mean square values were like 0.653, 0.94, and 0.27 for PDB dataset. Similarly, the other graph neural network and XG boost, called as XGraph boost for extraction features from molecules. So they used the dataset, the different datasets like ESOL, PDB. So all datasets were used. Now ESOL, for the ESOL dataset, the accuracy will the RMSE score was root mean square error score was 1.470 and 1.0236 and 0.329 respectively. Similarly, proposed another paper, they proposed a DL architecture called molecular bidirectional encoder representations for transformers. That is, BERT model was used a feature extractor for generating molecules, simplified molecular input line entry systems. 4 datasets were used for this valuation. This is ClinTox, TOX21, CIDR, and BBBP. The BERT achieved RMSE of 0.923 for ClinTox, 0.839 for TOX21, 0.875 for BBSP, and 0.695 for the CIDR. So similarly, another paper, they used, GNN, and RMSE was 0.695, and MPP, molecular property predictive approach. For this, the different datasets, pre-solve and malaria or CEP, lipophilicity, for all these things, RMSE squares were 1.22, 1.05, and 0.602 and 0.552 correspondingly. Super edge graph convolutional based requires attentional based DTA. So this extracted edge and node data from molecules. So they used the PDB dataset, and they were used, the PDBbind was used where SEGSA DTA achieved RMSE of 1.319. Another paper presented an architecture which combined graph attention network, GAT, and self-attention long short-term memory. So its RMSE value was 0.709 for GAT, and for lipophilicity, it was 0.885. Okay, so the different papers got the different methodologies they used on the different data sets, and RMSE values were varied. Further, it can also be seen that most of the work are utilizing BERT models for feature extraction and similarity scoring approaches. Okay, for evaluating the similarity among the molecules of protein ligands and its output given to the CNN for better outcomes. So this is the methodology. You could see the methodology there. Target protein ligand sequence, then the protein ligand 1, protein ligand 2, protein ligand 3, through the extraction EMSPLA, they merge embedding, amino acid level embeddings, protein level embeddings, chain embeddings, position embeddings. So we shall see the explanation of this particular diagram methodology. The proposed EMSPLA CNN architecture first evaluates the given protein ligand binding structure as given in the figure 1. The evaluation was done by evaluating the protein ligand sequences. Then the protein ligand sequences are separated using the BERT model for further evaluation using the proposed EMSPLA similarity evaluation. You could see there. So after the evaluation of the similarity, the sequence goes to the CNN. Okay, this is how it is going to the CNN. Okay, where it has two components, neural network layer and attention layer. Next, CNN is used as feature extractor, where the local features among the neighbor residues are extracted. Further, for extracting more features from the protein sequences, the CNN was stacked in blocks. You can see there. Blocks. Long sequences of protein and their binding residues were captured using the attention network. Finally, the outputs from both the network were merged, passed to the final connected layer, where the binary prediction took place using the classifier proposed in this work. The issue of overfitting was prevented by utilizing the dropout and way decaying approach. So this is not used in the convolutional layer. Okay. So this is about the methodology. If we want to discuss about the result and the discussion, the development of the protein ligand interaction prediction system was accomplished using the Python, by using the programming language called Python and C# programming languages, leveraging their respective strengths to create a robust and efficient solution. Python is known for its extensive libraries, frameworks, and data processing. Machine learning played a crucial role in this work. Libraries such as NumPy, Pandas, and SciPy facilitated data manipulation and analysis, while deep learning frameworks like TensorFlow and PyTorch were utilized for model learning and evaluation. Additionally, cheminformatics tools like RDKit were employed to handle the molecular data. On the other hand, C# was instrumental in integrating these models into larger software systems, ensuring efficient deployment and scalability through the .NET Core framework. C# libraries such as ML.NET and Ocar.NET were used for ML tasks and scientific computations within the application environment. The dual language approach, combining the Python's flexibility and comprehensive ML ecosystem with C# performance and integration capabilities, resulting in a powerful system for predicting protein ligand interactions. The hardware requirements included an Intel Core i7 processor, 16 GB of RAM, and at least 500 GB of free storage with a dedicated GPU recommended for DL tasks. The software requirements encompass Windows 11 alongside Python version 3.8 or higher, and .NET Core 3.1 and higher of C#. This comprehensive setup ensured that the system is both effective and scalable, capable of handling complex computational tasks in the realm of the protein ligand interactions. The evaluation of EMSPLA CNN was done using PDBbind dataset. This is the dataset we used. The PDBbind protein data bind v.2020 dataset includes binding site information for 19,443 complexes of protein ligand that have been identified through the experimentation. The information is available in both MOL2 and PDB file formats. The evaluation was done using the RMSE score. The results are shown in the figure 2. The figure 2 represents a comparison of various models, RMSE in predicting on their RMSE in predicting protein-ligand interactions, where a lower RMSE indicates better predictive accuracy. Among the models listed, the proposed EMSPLA CN stands out with the lowest RMSE root mean square error value of 0.67, suggesting it has the highest predictive accuracy. This model utilizes CNNs to capture complex spatial features of protein-ligand interactions, highlighting the effectiveness of the real techniques. SQM 2.20, another recent model from 2024, also demonstrates high accuracy with RMSE of 0.69, indicating significant improvements over earlier models. This is the data we get to know from the results and discussions. In contrast, Autotocvina, despite being a 2024 model, shows the highest RMSE of 2.85, indicating the lower predictive performance, which is due to its methodology. So other models like MSJG and SEGCAT-GTA show the papers we discussed in the literature survey show the moderate performance with the RMSEs of 1.27, 1.319 that you could see in the diagram, the figure 2. While IGN and RF score both from 2024 have slightly higher RMSEs of 1.82 and 1.89. So it is because of their methodologies that they have used. Deep DTA, a DL-based model, performs reasonably well with RMSE of 1.34, but does not match the accuracy of SQM 2.20 and EMSPLA CNN. These results indicate that newer models, especially those incorporating advanced ML and DL techniques, tend to have better predictive performance. The improvements seen in the models like SQM 2.20 and EMSPLA CNN underscore the importance of continued innovation in computational methodologies to enhance this accuracy and reliability of protein-ligand interaction predictions. So this is the graph that we could see, RMSE scores comparison with current state-of-art marks using the PDB dataset. So we are only concentrating on the PDB dataset. So MSGG, it is scored 1.27. SEGSA is 1.79. You have already, we already discussed regarding the years when they were deployed, and SQM 2.20 is 0.67. Autotocvina is 2.85, higher. So IGN is 1.82, RF score is 1.89, deep DTA is 1.34, and DSWA-CNN is 0.67. So RMSE scores achieved by the different models using the PDB dataset is shown in this figure 2. So finally, the conclusion. So in this work developed and evaluated the EMSPLA CNN novel protein-ligand feature extraction system that combines the CNNs and position-based attention mechanism. The EMSPLA was used for identifying the similarity between the proteins, and CNN was used to extract the local features from neighboring residues from the protein sequences. By leveraging the strengths of the Python and C#, the system benefited from the extensive ML libraries in Python and integration and performance capabilities of C#. EMSPLA approach utilized the PDB dataset and through the rigorous pre-processing and innovative modeling techniques, the EMSPLA achieved significant improvements in predictive accuracy as evidenced by an RMSE of 0.67. This is the result. EMSPLA CNN models' ability to capture both local and long-distance dependencies in the protein sequences, facilitated by the CNN and attention modules, underscores its robustness and effectiveness. So it has the robustness, it has the effectiveness, it has the accuracy. Additionally, EMSPLA contributes to precise structural similarity assessments, further enhancing the model's predictive performance. The results indicate that integrating advanced DL techniques within traditional sequence alignment methods can substantially improve the accuracy of protein-ligand interaction predictions. This work not only sets a new benchmark in a predictive performance, but also provides a scalable and efficient framework for future development in computational biology. Continued innovation in computational methodologies, as demonstrated here, is crucial for advancing the understanding and prediction of protein-ligand interactions. For the future work, the work will be extended for predicting and evaluating the protein-ligand binding affinity. Okay, thank you. So these are the references we refer. And I thank the International Journal, IJEECS. So this is about the paper that we have submitted to your journal. So thanks to the Master Program of Electrical Engineering and universities Ahmad Alam UAD. I thank ESPCRG and I thank IAES. Thank you so much. Thanks for giving the opportunity.